Objective, students will be able to describe the structural differences between viruses and cells. So, if you remember last week during the ACT testing, I pushed out kind of the last assignment, which was going to be that virus thing. Um, we didn't. It was kind of last minute, so I didn't get a chance to double check it. So there were a lot of things I didn't like about it. So we're going to redo that today. If you did it, that's fine. I might even give you some extra credit for the test. But we're going to redo the assignment because there a lot of it was just kind of nonsensical. Normally, whenever we got to viruses, we would have covered a lot of that information, but because we haven't, we're kind of doing things out of order this year. A lot of the information on there was not stuff you needed to know for the test. So I wanted to go back and make sure that I simplified it. I made it just test material. So the assigner stuff I have is the IP. It's also the GP viruses. We're going to get through about 75% of it today if I can kind of zip through everything. But yeah, viruses. Today's going to be the last day of testable material. So viruses will be on the test, but only a couple questions. It's not too bad. But yeah, we do have our test tomorrow. Tomorrow we have the essay portion. That means that in the morning, or sorry, in the beginning of class, we'll have something else to do. And then after that, we'll get to our essay, probably the last 20 or 30 minutes. Then the following day on Thursday, I'm sorry, Wednesday, we'll have our multiple choice exam.
Uh,
Okay.
Mr. Raymond,
Mm-hmm.
will there be like a hoot game if we have a test tomorrow?
That's the plan.
Okay.
All right, so here's our do now. I'm going to go ahead and do 3 minutes, try and save some time. I'm looking for an incorrect answer. Pick an incorrect answer and explain why it's incorrect in the chat box. Okay, I'm going back to the chat. So let's see. Afternoon, Kaylee. I saw you turn on camera earlier. That was fun. Yeah, Hugo. There's no food to bring you. Sorry about that. Justina's focused. I like it. Josiah is calm. Kaylee green at ease.
I'm
Jacob
happy.
taking it easy. Casey still getting that virus thing. And rise and shine. That's funny because people were sleepy. Afternoon, Faye. Caitlin, somebody else was shouting they were calm earlier, so they're trying to steal your thunder. Hi, Grace.
Um,
Hi,
Mr. Raymond,
Gigi. Mm-hmm.
um, I think this incorrect answer for me would be J.
Okay. But I want to know why it's incorrect. So you're going to pick one. Tell me why it's wrong.
Okay.
Well, here, let's wait for the timer to go off first.
Uh,
Gigi, thank you. Casey, hello. I said Casey already. Amian, nice and calm. All right, now we're getting to answers. Let's see. Davian says that F is incorrect because chloroplasts don't remove waste and mitochondria produce ATP. Very good, Davian. Give you a thumbs up. Casey, red, because she's always mad. Whenever you look at her, she's just so angry all the time, just like the angriest person I know. Grace says she's tired. I agree with you, Grace. I'm tired too. Josiah said F is an incorrect statement because it says that the mitochondria produces sugar. Very good. Good job, Josiah. Also gonna give a thumbs up. Hugo, a lot of J's. G. Ooh. You don't get it? Like the right answer or the wrong answer? It's okay. All right, I didn't get to read all of them, so I'm just gonna call on some people. Brighton, why don't you tell me what you put down? Okay, so that Brighton did. How about Timothy, if you're there, would you mind explaining your answer?
I can explain my answer.
Go for it, Brendan.
Um, I think the answer is J because for the chloroplast, I don't think it converts energy.
Okay, what else?
Um, that mental coordination, um, it only stores water, but it doesn't produce sugar.
Well, be careful because the mitochondria is the powerhouse of the cell, right? So the mitochondria produces energy. It's one of those energy converters. But you're right, it does not produce sugar, which is why that one is wrong. So very good. All right, we're going to go ahead and move past it because, like I said, we're running short on time. H was the correct answer, so this is the one that pairs everything correctly. So vacuole stores water, chloroplast produces sugar, mitochondria converts energy, and the cell membrane regulates what comes in and out of the cell. So I just wanted to see if you knew how well these were paired up, so you could kind of pick out which ones were wrong. I wanted to stress one more time, here we're looking at a plant cell. Can I get somebody to read this green text over here on the right? Bless you.
I can read it.
Go for it.
The chloroplast is in plant cells and is responsible for photosynthesis. It's not found in animal cells, and animal cells also lack the cell wall that gives plants shape and support.
Very good. Thank you. And I saw some more in the chat. Thank you, Eileen. Yeah. So just wanted to make sure that we saw it one more time. Chloroplast is the one that's responsible for photosynthesis. That's where it said is producing sugar. We haven't really gone over photosynthesis a whole lot, but when you think about fruit, think about plants, right? So plants produce, they take in sunlight, they drink water, they breathe in carbon dioxide, and then as a byproduct of that, they make sugar, usually in the form of fruits, and then oxygen that we breathe. So the chloroplasts are responsible for photosynthesis. That's where we're talking about producing sugar. But the chloroplast in the cell wall, not found in animal cells. And remember that the cell wall gives the plant shape and support. All right. Any questions about that? Pretty straightforward, right? Not too hard. Okay, let's move on. This is one of the questions from the exit ticket last week. It was pretty good. 58% is not bad, but I thought we could do a lot better. Does anybody want to tell me why number 4, the answer was that last one?
What does the last one say?
Well, I've heard from you already, Brandon. How about somebody else help us out?
Can I help out?
Go for it.
It's wrong because the first two are true because those are both two factors that support the fact. So if we're looking for one that's not, if we know the first two are correct, then that only leaves the last one to be false.
Very good. So again, just like Brighton said, we're looking for the incorrect one, like which one does not support the endosymbiotic theory. Remember, the endosymbiotic theory is about the prokaryote invading other prokaryote, and that's where you get eukaryotes. So mitochondria and chloroplasts have their own DNA. That's true. That's part of the reason why we know that the endosymbiotic theory is probably true. And then mitochondria and chloroplast reproduce like bacteria. Also true. So that's support and evidence used to support the endosymbiotic theory. Mitochondria and chloroplast both lack membranes. We know that is definitely not true. And that's in fact, the fact that they have membranes is more proof that the endosymbiotic theory is true. So can I get somebody to read this little bit down here at the bottom?
I can read it, sir.
Go for it.
The membranes are used as evidence to prove that endosymbiotic theory. Prokaryotes were not digested and lived in symbiosis, the organs of eukaryotes.
Very good, right? So this is where we get the origins of eukaryotes. A long time ago, these mitochondria and chloroplasts were bacteria that didn't get digested. Instead, they evolved some kind of symbiotic relationship, and now we have eukaryotes. So remember, there were prokaryotes first, then because of this endosymbiotic theory, now we have eukaryotes. So the prokaryotes were the kind of predecessors to the eukaryotes. Okay, exam. So we have our essay tomorrow. Our multiple choice is going to be on the 21st. So I want to make sure I touch on that one more time. Now, I had some questions about tutorial. I went ahead and decided I'm going to do something very similar to what Ms. Jacobo does. So she usually has that tutorial time from like 5 to 6. I'm not quite that brave. So what I'm going to do is I'm going to do 4:30 to 5:30. Definitely optional. You don't have to go. I don't know if I'm going to give any extra credit quite yet. I'm not saying that I won't. I'm just saying that I haven't decided quite yet. But I wanted to make sure that I had some extra time because we did lose a day from ACT testing. So I know that some people still feel a little bit uncertain about what's going to happen. I know it's not quite in time for the essay, but it will be in time for the multiple choice. So if you want to, it'll be tomorrow, 4:30 to 5:30. So just after school. Okay. I'll probably have the teams going, and then I'll just talk about the material, give you a few practice questions, and that'll be it. Again, nothing too serious. It's just really to help you because we did lose some time this week. Okay, any questions about that? I'm going to guess that's a no. And let's keep going. Okay, can I get somebody to read the objective, please? Go for it, Justine. Yeah. Well, then they can't hear you.
Students will be able to describe the structural differences between viruses and cells.
Thank you. See, the world didn't end. You're still fine. Everybody's still great. Right? I don't know why y'all are so embarrassed. Anyway, thank you, Justine. So we're looking at viruses. We're not going to get too in depth with them because later on, like I mentioned, we're going to have an infection unit, like with bacteria and viruses, and we'll talk more about their life cycle then. But for the time being, we're just kind of looking at their structure and kind of comparing viruses to cells. So, like I said, I went ahead and updated this sheet from last week. So if you did it, that's great. You can probably copy and paste most of your responses over. All I really did was edit it to make sure that it's more focused on the stuff that's going to be on the test. Before we get to that, though, I wanted to run through that PowerPoint because I did the same thing with that. Boop. Yeah, so I do take responsibility for that. I wish that I would have had more time to review the materials before I pushed them out. Everything was just so chaotic last week. I didn't really have a lot of time. Okay, so we're going to look at viruses just real quick and kind of compare them to cells. Now, can I get somebody to read this slide for us?
What are viruses?
Mm-hmm.
Okay. Tiny organic parasites smaller than bacteria that can cause disease. They are non-living things because they need a host cell in order to reproduce. Viruses usually have three main parts. Number one, a protein shell called a capsid. Number two, a genetic material, either DNA or RNA inside the capsid. And number three, surface membrane proteins called projections for the virus attached to the host cell.
Very good. So viruses are not alive. The reason is because they can't survive on their own. It's not because there's some kind of bias to things that can't reproduce, but if they can't reproduce by themselves, then they can't really live, right? So they need a host to survive. They're essentially organic parasites, right? We're talking about parasitic relationships earlier on. It's hard to say whether or not viruses benefit because all they're really doing is using the host to reproduce. There's no kind of like other benefit besides that, but they are using it. So one of the creatures is hurting from that relationship. And then the three things, the capsid, which is the protein coating. So if you look over on our diagram, you see the capsid is surrounding the genetic material. So it's acting kind of like a cell membrane a little bit, but it's not. It's capsid, it's protein coat. They also have genetic material. So just like everything else that exists, they have DNA or RNA. And then they have little cell surface projections, which are used for attachment. They're called cell surface proteins or projections, whatever. They all kind of look the same. They all have those little projections. The reason is, think of it like an ID tag. So, like when you say, like, my name is Steve, you got a sticker that you put on your chest. It's kind of like that. The virus is saying, hey, look, my name is Steve. I'm a great guy. Let me in, right? So it's using it to kind of gain entry into other cells. Okay? So the capsid made a protein. nucleic acid, DNA or RNA. Envelopes we can get to later. They're actually kind of tricky. What happens is that when the virus comes inside, it wraps itself around the cell membrane. So it's kind of like a wolf in sheep's clothing. As they enter the cell, they're literally dragging a piece of the cell membrane inside. So it's like hidden. That's one of the ways HIV works, makes it dangerous. So that's an envelope. And then, of course, those surface membrane proteins are used for attachment, so that the virus can attach to the host. Okay, so they are similar to living things, but they are not living things because, again, they can't reproduce on their own. So they have those nucleic acid, they have proteins, and they don't have any membrane-bound organelles, just like bacteria. They're very simple, so they don't have any kind of organelles. You have bacteriophages. Those are viruses that affect viruses. And then you have regular old viruses there on the right. Okay. So can someone tell me what all these viruses have in common?
Hey, Mr. Raymond, I just want to ask,
They can all affect you in a very horrible way, and like they can affect your or organs and cell systems that can possibly
good.
potentially kill you.
That's true. What were you saying, Brandon?
I just want to ask, in the middle, what kind of bug is that?
That's not a bug. That's a bacteriophage. So bacteriophages are viruses that infect bacteria. So when we think of like COVID, right? COVID is a virus. We think of that virus infecting us, but that virus wouldn't infect a bacteria. Conversely, that bacteriophage wouldn't infect us because they would have different cell surface receptors. So those projections wouldn't be able to get into our cells. So they have to be specific to certain creatures. So the bacteriophage is specific to bacteria. They look crazy though, huh?
Mm-hmm.
All right,
In the
Josiah was
green and blue
very
one,
good.
I have a
Josiah
question.
said they're not alive.
Question.
Uh-huh.
In the green and blue one, what are the little like antennas sticking out? Like, what is your purpose?
What's the purpose of what?
Those are the like little antennas or whatever there are. Like
Yeah,
sticking.
they're kind of like antennas. They're used to attach to a host. So these, remember, these are like cells, right? They're not cells, but they are cells. The reason they're not cells is because they don't have like that membrane around them. They use those projections to attach to things. So think of it kind of like an arm. Like when it comes close to one of the cells, it like attaches. When we talk more about viruses later during viral reproduction, you'll see. But for now, I just wanted you to be able to recognize the shape. Like you're looking at these common shapes, they all have these surface projections because the viruses need to have a host. So they
Oh,
have
so
to have a
kind
way to attach.
so that's kind of like Velcro.
Yeah, kind of like Velcro. It does look like a spider, doesn't it? It looks weird. What's funny is to think that this is not made up from some comic book artist, right? Some guy didn't dream this up. This is in biological nature, right? This was made not by human hands. It's pretty crazy. Very good, Eileen. They all have genetic material. You guys are doing good. So they don't have any organelles, just like bacteria. They're not alive. They have that capsid, which is here in the bacteriophage. It's basically what's covering the genetic material. And then pins are projections to attach to a host cell. So here are the projections on that virus. These legs on the bacteriophage are used to kind of anchor them to bacteria. Pretty cool. All right, that's it. So like I said, it's not too complicated. We're just looking at the structure for these viruses. I don't want to overwhelm you guys, especially right before the test. But if we're talking about cells, we've talked about bacteria and eukaryotes. I wanted to make sure that we also touched on viruses. But we'll talk more about viruses a lot later because they're really interesting. Oh, whoops, I just shared the same thing. Oh, yeah? Who's it from? Okay. Yeah, if we had more time, I would love to really dig into viruses, but we'll get into it later. Okay, so here are the questions on the sheet. So if you have your guided practice up, you can go ahead and start filling it in. So what are viruses? They're tiny parasites that can cause disease. Are they considered to be alive? No, because they need a host to reproduce. The parts of a virus, we got that protein coat or capsid that surrounds the genetic material and the projections attached to a host cell. And then of course the function of those surface membrane proteins are to allow the virus to attach to the host cell. So sometimes they're called projections, sometimes they're called surface membrane proteins. Really, it's just the thing that's on the outside of the virus that helps it to attach to the host. Because again, when you think about these things in nature, they don't have arms and fingers like we do, right? They have different structures, so they have those projections that helps them attach to things. Okay. Any questions about that? I guess that's a no. Like I said, we're going to make up for some lost time, so I am going to move a little faster than I normally would. Okay, here's the next one. This is number 5. What you're doing is you're classifying these as eukaryotes, prokaryotes, or both. So, would someone mind coming off mute and explaining why I said these are eukaryotes?
I volunteer as a tribute.
Go for it.
The reason you put eukaryote as a nucleus is because it carries a nucleus, right?
Very good.
The reason you put plants, animals, and fungi is because those have a nucleus.
Very good.
And
10 times bigger is because they have more organelles and they have more organelles.
Isn't it the vacuole also like 10 times bigger or something like that? Which one was one of those in plants that are 10 times bigger?
That's why he put plants, animals, and fungi.
Right, so very good. Hugo kind of zoned in on it. They are more complex, which is why they have many different organelles. It's also why they're bigger than prokaryotes. They cover plants, animals, and fungi. And of course, the biggest distinction is that eukaryotes have a nucleus, while prokaryotes do not. All right, so here we're looking at prokaryotes, very, very more specific, right? Prokaryotes, again, no nucleus. Pro rhymes with no, right? So no nucleus. An example of a prokaryote would be bacteria. And of course, they are very, very tiny. If you remember from the video a while back, that bacteria was like a little tiny, and the eukaryote was real massive, right? Okay, and finally, both. Would somebody mind coming off mute and explaining why these are both?
What do you mean by both?
So these are characteristics shared by both prokaryotes and eukaryotes.
Okay.
Because they both have ribosomes.
Yeah.
Very good.
They both create DNA.
They both have DNA.
They both have a cell membrane.
That's true. And lastly?
They both have a cell wall.
Very good. Thank you, Gabriel. And like you were saying a second ago, which eukaryotic cell has a cell wall?
The plant cells?
The plant.
Thank you, Brighton. Right. So remember that there are animal cells and plant cells. Plant cells are the ones with the cell wall. So very good. All the guys are doing great. Okay, so there you go. That was pretty simple, right? Pretty painless. Now, on the other worksheet, it had like questions about which one of these elements are most common, and I think stuff about the macromolecules. We haven't quite gotten to that yet, so that's why I took all that stuff out. So don't worry about that. Okay, so I don't have time to do more. I was going to go back and do some more, but we don't have time for it. I wanted to make sure that I gave you time for your independent practice. So we've been talking about the endosymbiotic theory for a couple days now. What I want you to do now, we've talked about the specific lines of evidence. We've talked about what the theory is itself. What I want you to do now is to combine that into your own thinking. So in your own words, I want you to describe the endosymbiotic theory and the evidence that supports it. I went ahead and gave you some vocab terms that you can use. You don't have to use them all. I just put them there if you wanted them. But what I'm looking for is for you to explain, what your thinking is about the endosymbiotic theory. This was very similar to the eggs ticket question a while back, so I wanted to make sure I asked it one more time so that you can really, really hone in your thoughts about it. If you don't know, you have time to kind of refresh your memory. You can always ask for some help. But remember that I am taking your answers in the chat box as part of your participation grade. So it's your do now, your independent practice, and your exit ticket, which is going to your performance. Okay? Are there any questions about what I'm looking for here with your independent practice? Did I misspell it? Oh, okay. Thank you. Okay, so I'm going to give you, let's see, how much time do we have? Yeah, we got time. I'm going to give you 10 minutes. I want you to, again, write down. I'm not going to put like a limit as far as how many sentences. What I'm looking for is that you understand the theory, maybe throw in some vocab words, show me that you understand what this is and what the evidence is for it. I didn't want to make it a CER because we did that last week and it was very similar to the one on your exit ticket. So this one, I'm just looking for a flow, like just write. I'm looking for you to explain it just in your own words. If you want to make it a CER, that's great. The claim could be that, you know, the endosymbiotic theory is the origin of eukaryotes, and then you can use some evidence and back it up with some reasoning. If you want. I want it to be a little bit more flexible. Okay, so 10 minutes. Let's make it 9 minutes now. Here we go.
Sir, do we post it here? Do we put it here in the chat box or in the thing that says independent purpose in the assignment?
Go ahead and put it here in the chat box.
Okay.
Eileen just sent me a great message connecting the dots between eukaryotes and the endosymbiotic theory. So thank you for that. And Gabriel, that's good, but you don't define what the endosymbiotic theory is. I guess you do. I guess you do. Yeah, actually, you know what? I take it back. That is a good response. It's very succinct. And Hugo, I think what you're trying to say is that it's called photosynthesis, but the organelle is the chloroplast. Very good, Grace. The only thing I want to clear up is that the prokaryotic cell was engulfed, like it was kind of eaten by another prokaryotic cell. It's okay, Hugo. You guys doing great. Good job. Yeah, I think I'll go ahead and read what Eileen sent me because I thought it was really cool that she made that connection. She said, is the reason why eukaryotes are larger and more complex than prokaryotes because of the prokaryotes that engulf the other bacteria that had other properties. So I think what she's getting at is that, you know, that one ate the other one. So the reason the prokaryote is so small is because the eukaryote time ago ate a prokaryote, and so over time it got bigger and bigger and more complex. So yeah, it's a very good observation.
When school isn't a place you have to be,
Oh, actually, very good, Clint. I was actually going to show this video later. So when we get to our infection unit, I was actually planning on showing this. So yeah, if you guys want to watch the video that Clint posted, I'm definitely going to show it later. It's one of my favorite videos from Kirkusat. But a little bit too in-depth for what we're doing right now. Thank you, Jay. It's very good. Thank you, Alan.
Mr. Raymond,
Mm-hmm.
mind if I just tell you?
Let's wait until the timer's over. Is the timer almost over?
No, it's still not yet.
Yeah, let's give it a little more time. Ooh, very thorough, Brayden, but I like it. It's good. And Hamza, you're on the right track. It's a little confusing, but I think I understand what you're trying to say. Yeah, Jay, I think this is how it's going to be from now on. Eileen, very good, seeing that you're repeating what you said earlier. Very good, Kiki. And Timothy, I think you'd be a little more specific. Oh, Karina, good job throwing symbiosis in there. I like it. Thank you, Tatiana. Ooh, Josiah going deep. I like it. That's great. Very good, Cheyenne. I hope that means this is a little more clear to you than it was before. Very good, Destiny. Destiny's bringing in that that the chloroplast and the mitochondria. Very good, Destiny. Thank you, Lauren. Good job. Thank you, Giselle. Good job, Faye. Bringing in the energy. I like it. Good job. I like that. Thank you, Amian. Good job, Gigi. Thank you, Mr. JJ. Okay, I almost got through all of them. Like I said, I am going to be reading these and putting it as your daily grade, so thank you guys for submitting it. You guys are really doing some great responses. Thank you, Casey, for doing a CER anyway. I thought I'd be giving you a break, but you just went for it. And thank you guys for being really in-depth. I do appreciate that. Like I said, the CER is very important, but I wanted to make sure that, you know, I wanted to give you a little bit of freedom this time around just to kind of express yourself. But yeah, I'm very happy with what I've been seeing with the CERs and what you guys are doing here, so really, really good job. Okay, let's see how much time we got left. Yeah, perfect. I wanted to make sure that I'll have plenty of time for the exit ticket today because it is a little bit longer than normal. The reason is because I wanted it to be like a little mini review for the test coming up. So like I said, the test tomorrow is going to be your essay portion. After that's going to be your multiple choice. So the questions that I'm asking on this review are relatively simple, but they do cover everything. So from the organelles to endosymbiotic theory to viruses. So I want to make sure they give you plenty of time. And like I told you the other day, I'm going to go ahead and encourage you to keep going until you get 100. I know it's a little bit more work for me, that's fine, but I want to make sure that you guys are utilizing this as a way to like really get to mastery, to make sure that you can show me that you understand and that you know what it is that's going to be expected of you on the test coming up. Okay, so again, essay test tomorrow, multiple choice day after that. I will have that optional. Let me go back one more time so you can see it one more time. I will have that optional tutorial time tomorrow, 4:30 to 5:30. Again, not required, totally optional, but it is designed to help you, give us one more chance because we did miss that one day from ACT testing. If you have questions, make sure that you bring them in. That way we can do our best and utilize our time as widely as possible. Okay, yeah, we've got our 15 minutes for our exit ticket, which is perfect because it might take a little bit. Are there any questions before we get to it?
No questions.
Thank you, Jay. If you have finished your exit ticket, that's great. Make sure that you go back and look at your CK through 12. If you had got a chance to write it down earlier with your assign yourself. So 7.8, 7.9, 7.10. So if you finish your exit tickets, you have those CK through 12 assignments to go ahead and start reading. It'll definitely help you for the test. Okay, then let's go ahead and jump on into the exit ticket. Good job, you guys. And thank you guys for asking questions in the chat messages. If you have any questions, please let me know. I'll answer them right now. Remember, it's a new 9 weeks, another chance to be great. And don't forget, if you get a message from me this morning that you did not complete your exit ticket last week, please get on that. That's fine, Casey. And good job, Jay. Like I said, go ahead and make sure that you go back to your CK through 12.
I missed one,
That's still pretty good.
which was a pick.
Oh, select all that apply.
Yeah, select that.
That's tricky. Like I said, if you want to go back and redo it, you definitely can. Ooh, Cameron, very good job. I'm going back and reading the responses while y'all are working on your next ticket. Good job, Jacob. Hey, good job, Justine. Very simple. Nice. Way to go, Davian. Good job. Oh, you're guessing on some of these. Again, if you don't get 100, I really encourage you to go back, see what you missed, try and figure out why you missed it. If you don't understand it, please come to the tutorial tomorrow, 4:30 to 5:30. So go ahead.
Well, Mr. Raymond, well, I didn't get 100 for the first time, but I did get one for the second time.
Good job. Thank you, Cheyenne. Cheyenne, send me a quick message to let me know how you're feeling. I know that you were feeling a little overwhelmed a while ago. Good job, Grace. You too, Faye. And again, if you're finished, make sure you go back. You've got your CK through 12. Way to go, Gigi. Good job. And Hugo, yeah, I bet. Very convenient. Yeah, you guys are doing great. Some of y'all are guessing, I can see. But for the most part, doing very good. Good job. Keep up the good work. Make sure you're watching on number 6. There's more than one answer. Yeah, Clint, like I said, I encourage you guys to go back and get 100. If you feel like you really know this or if you're struggling, either way, I want you to go back and do as best as you can. Yeah, same with you, Cheyenne. Okay, Brian, 1 second. And sent. It should show up as a private message for you. Okay, sent. Thank you, Josiah. Way to go, man. Amian. Amian. That's okay, Eileen. I know it's you. Love the positivity. Thank you guys for congratulating others. Oh, I can probably stop.